hear some of this information again, it won't hurt. Where we left off, though, was with mitochondria. And remember, we talked about two membranes, and particularly that inner membrane having a lot of folds in it, those cristae. And that's the way the mitochondria can increase the surface area so that it can have more room for those membrane-bound enzymes to be able to synthesize ATP. So more sets of those membrane-bound enzymes working to synthesize more ATP at the same time. And inside that inner membrane, you've got this internal fluid-filled space called the mitochondrial matrix, and that's where the mitochondria has its own DNA, ribosomes, and enzymes. So again, the function of the mitochondria is cellular respiration, with the goal of generating ATP, and this comes from the breakdown of sugars, fats, and other fuels. Sugars are the most direct route to get that ATP, but the cell can use fats and fuels by doing some additional chemistry first before sending the sugars to the mitochondria. This has to happen in the presence of oxygen. There are other anaerobic options for the cell when there isn't oxygen present, and again, we'll talk about those later. But this thing that the mitochondria does to create ATP requires oxygen. That breakdown of larger molecules into smaller molecules in order to generate energy is generally called catabolism. Specifically, when the cell is generating energy in the presence of O2, that's aerobic respiration. And here's this is the same diagram of a mitochondria that was on the last couple of slides, but this shows you what the mitochondria looks like under a microscope. So you see kind of one big, fat, larger, whiter mitochondria with the cristae, lots of those folds there. And then in the top right corner, you see some smaller mitochondria, some of them longer, some of them shorter. So they vary some in size in reality. And I mentioned those membrane-bound enzymes, and in reality, they are a complex of enzymes to run this reaction. And by putting them in that membrane near each other, it helps the reaction move forward more efficiently and therefore faster, because as the product of one reaction moves to the enzyme that catalyzes the next reaction, that enzyme is right there nearby. And so then you can see the yellow arrows as those products are moved through the enzymes in the reaction. If they're nearby, that can happen a lot faster and a lot more efficiently. And then again, also because you've got the folds in the membrane, you can have a lot of these enzyme sets, these enzyme complexes in there running the reaction simultaneously. So mitochondria can divide themselves. We'll talk about this in reference to the endosymbiotic theory in a bit. But you can see, if you look on the left, that's some mitochondria dividing. And then on the right, that's a bacterial cell dividing, so a prokaryotic cell. And they look very, very similar in terms of how it happens. So if you look at the details of bacterial cells and how they do binary fission, which is their cell division, and if you look at how mitochondria does that, the process is very similar. Mitochondria are very important. Almost all eukaryotic cells have mitochondria. Some cells have one very large mitochondrion, so that's the singular of mitochondria. Or there could be hundreds to thousands of individual mitochondria in a given cell. And really the number of mitochondria depends on how much aerobic metabolic activity that particular cell is doing. So depending on its job, it may need to do more or less aerobic metabolic activity. And the more activity it's doing, the more energy that cell needs, and therefore the more mitochondria it's going to have. And so a great example of a cell that would have more mitochondria would be your muscle cells. Your muscle cells have to do aerobic metabolic activity all the time. Even if you're just walking between classes, that's aerobic metabolic activity. And so your muscle cells are going to have more mitochondria than some other cells in your body. And depending on how much aerobic metabolic activity you engage in, you may have more or less in your muscle cells versus, say, a classmate of yours who maybe does more endurance sports versus another one that tends to be much more sedentary, is going to have fewer mitochondria even within the same cell type. And again, mitochondria are pretty much all eukaryotic cells have mitochondria, and it is the same thing no matter the type of cell. So animal cells have mitochondria, plant cells have mitochondria, because even though plant cells have the chloroplasts that are taking the sun energy, doing photosynthesis, once that photosynthesis is done, the cell still has to be able to take any glucose that was made as part of the process in order to be stored and then use it to power the plant cells' functions. Okay, so let's talk a little bit about some things that are common to mitochondria and chloroplasts, and then we'll transition into talking about chloroplasts. They both are involved in transforming energy and generating ATP. The chloroplast certainly does generate ATP as part of photosynthesis. Often it will then take that ATP and convert it further into glucose or other macromolecules. But for mitochondria, you're taking that glucose and generating ATP from there. They both have double membranes, so those two layers of membrane. And they're both what are called semi-autonomous organelles, so they can move independently, they can change shape, they can divide. And they have their own internal ribosomes, their own DNA, and their own enzymes. So chloroplasts, these are organelles that are specific to plant cells. Animal cells don't have chloroplasts. There are some kind of unique, weird cases of some animals, some organisms in the kingdom Animalia who go around, eat algae, take their chloroplasts out and sort of use them for their own purposes, kind of hijacking the chloroplast, but they don't have their own chloroplasts. They're just using them. Plants are the only ones that actually have the chloroplasts themselves. Chloroplasts belong in a class of plant structures called the plastids. There's a lot of different kinds of plastids, but just kind of some examples. One you already looked at, amyloplasts. So these are the storage, these are structures for the purposes of storing starch in roots and tubers. So we were looking at those in the bananas. Chromoplasts store pigments for fruits and flowers. So we talked about storing pigment molecules in the central vacuole, and that does also happen. But the chromoplasts do this as well in plant cells that need to have some kind of color. And then the chloroplasts, they store chlorophyll and then function in photosynthesis. And chlorophyll is actually also a pigment molecule. It just happens to also have this other function in photosynthesis. In leaves, so the chloroplasts are specifically in leaves and other green structures of the plants and algae. Okay, so again, chloroplast, two membranes. And inside of that internal membrane of the chloroplast, you've got what's called the stroma. Inside the stroma, just like the mitochondria, you've got DNA, ribosomes, and enzymes. If you look at that diagram, you kind of got those green coin-shaped stacks. And those membranous sacs in the stack are called the thylakoids, and that's where the ATP is made. The whole stack of the thylakoids is called the grana. And again, we'll get into more detail about both the mitochondria and the chloroplast and how exactly this works in the next unit. And just like the mitochondria, again, why they have these internal sac-like membranes and they have them stacked up like that is for the purposes of increasing the surface area for those membrane-bound enzymes. And so you've got more surface area on those membranes to run more reactions. Okay, and so again, chloroplast, the function is photosynthesis, generating ATP, and then synthesizing sugars. And the chloroplast takes solar energy, converts it into chemical energy, either in the ATP or the sugar, and then if it's going to be specifically sugar that's made as the end product of photosynthesis, because we'll talk about this, photosynthesis can potentially stop or reroute at different places, but kind of keeping the simple case where you're going towards sugar, the sugar will be made from CO2 and water. It's just the solar energy is the energy to catalyze that reaction of putting the CO2 and the water molecules together. Chloroplasts are also semi-autonomous, so they can move, they can change their shape, and they can divide independently. And just like mitochondria and bacteria, they can reproduce by pinching in two. So unlike our other organelles in the cell, where some other part of the cell has to make another one or make more of that organelle, chloroplasts and mitochondria can reproduce on their own. Okay. And again, the membrane-bound enzymes in those thylakoid membranes form complexes, just like they do in the mitochondria, where you have all of the enzymes necessary to run the reactions of photosynthesis all right there near each other, co-localized in that membrane in order to increase the efficiency and speed of the reactions. Okay, so mitochondria and chloroplasts similar to each other, but they're different from other parts of the cell, different from other organelles. So they are not part of the endomembrane system. And the endomembrane system, when we talk about the endomembrane system, we mean the nucleus moving out further into the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi, the vesicles, all of those organelles that have membranes and sort of flow from one into the next. They can grow and reproduce on their own. So again, they're semi-autonomous. They can do some of these things on their own. Generally, their proteins that they use are going to come still from the free ribosomes and the cytosol that then come in. The protein gets made out in the cytosol and then comes into the chloroplast for the mitochondria. But they do have a few ribosomes of their own, and then they have their own circular chromosome that helps direct the synthesis of proteins that are produced by their own internal ribosomes. So they do some amount of their own protein synthesis. Bacteria, so this prokaryotic cell down here, also has circular chromosomes. If you think about animal and plant cells, something we actually haven't talked about yet is that they've got linear or straight chromosomes that are double-stranded DNA, just long strands. But bacteria, their chromosome is actually circular. And that's the same thing with mitochondria and chloroplasts. So even though they have their own DNA, it's more similar to bacterial chromosomes, bacterial DNA, than it is to eukaryotic DNA. Okay, sorry. Let's talk about the evolution of the first eukaryotes, because the oldest life on Earth would have been much more similar to the bacteria that we see today, where they don't have the endomembrane system, they don't have some of the other organelles like the mitochondria and the chloroplast. And so there was a gradual evolution of some of those cells into the first eukaryotes. Now, we'll talk about this in evolution. That doesn't mean that prokaryotes haven't evolved since then, but they just have evolved on separate tracks. So the first thing that happened in the development of eukaryotes is the development of internal membranes. So ancestral cells still had that cell membrane on the outside, but then at some point, some of these first or proto-eukaryotes started to develop membranes on the inside that helped them create these internal microenvironments where they could have some specialization happening inside the cell that helped increase their efficiency. So it helped some chemical reactions happen closer to each other, or helped the cell separate some chemical reactions that are maybe harmful to the cell, lots of things like that. And so then the cells that had developed these internal membranes would have some kind of advantage in terms of survival, maybe even reproduction. And then that would lead to natural selection because they'd be the ones that'd be more likely to survive and reproduce. And all of this happened somewhere about 2 billion years ago. Okay, then after that point, the first endosymbiosis happened. And all of these years, these estimates, some of this is based on fossil evidence. Believe it or not, they have found fossils of individual cells over time, but a lot of it is just estimates based on how long things would have taken to have happened. And so some indirect evidence. So time is, some of the time here is estimates. But the first endosymbiotic event happened, and it is believed that the mitochondria entered the cell first. And how this happened is basically some ancestral eukaryotic cell engulfed or took in anaerobic bacteria, but for whatever reason didn't digest them. So maybe the lysosome malfunctioned or something happened where that, what would normally happen when a cell takes in a bacteria, didn't happen. And what developed was a mutually beneficial relationship. So the ancestral eukaryotic cell benefited because this aerobic bacteria was producing lots of energy, enough energy in excess that the ancestral eukaryote could get some. And the aerobic bacteria benefited because it got some of the nutrients and resources from the ancestral eukaryote to be able to do its metabolic activities, and then also protection from environmental hazards. And so then, because this relationship was beneficial, it helped this ancestral eukaryotic cell that entered into this relationship be more likely to survive. And again, natural selection happens, and so it became much more common. Then the second endosymbiotic event happened, and this is where chloroplasts enter the picture. And the reason scientists are reasonably sure that mitochondria came first and chloroplasts second is that almost all cells have mitochondria, but only plant cells have chloroplasts. And so it wouldn't make sense for the chloroplast to have come first in the ancestral eukaryote, then the mitochondria, and then later animal cells to then have lost the chloroplast and plant cells to have kept it. It makes more sense that the ancestor of all eukaryotic cells took in the mitochondria, and then the ancestral plant cell took in the chloroplast, while some other ancestral eukaryotes that eventually became animal cells did not. And that was the splitting event. And that's, again, it's just the hypothesis is that that's how it happened, because that would be the simplest explanation for what we observe now. But how the chloroplast got there, in any case, was that this ancestral eukaryotic cell engulfed some kind of photosynthetic bacteria, but again, didn't digest it for whatever reason. And this led to a mutually beneficial relationship where, similar to the mitochondria, the chloroplast, this photosynthetic bacteria, was getting protection from the environment, access to additional nutrients from the eukaryotic cell, and then the eukaryotic cell was getting the excess energy from the chloroplast beyond what it was producing for its own needs. And so both benefited from this relationship. So let's talk about the summary of evidence for this endosymbiotic theory. And you see here the pictures of Lynn Margulis from, and you read about her in the reading. And she was really instrumental in collecting the evidence for this theory to give it full support. And so the first general line of evidence is structural. Mitochondria and chloroplast resemble bacteria in a structural sense. Genetically, mitochondria and chloroplasts have their own circular DNA, like bacteria. And the fact that they have their own DNA in the first place, compared to other organelles, which do not. And then finally, the third general category of evidence would be functional. So mitochondria and chloroplasts can move freely within the cell, and mitochondria and chloroplasts can reproduce independently from the cell. I'm not going to play this video kind of summarizing the ins and outs of endosymbiosis, but if you were, after the reading, after that brief set of notes, still either kind of curious about it or not sure you understand how this came about or the lines of evidence, you can certainly check out this video and it may help. All right, well, that's it for the notes.